Hello, welcome to Merritt Bioscience. I'm standing in front of Building S up here on Merritt College campus in Oakland, California. In the basement of this Barbara Lee Science Allied Health Center building is a microscopy center. And today I'm going to take you down into the lab and we're going to work on some microscopes. Welcome to Spring 2021. Hello, welcome to Bioscience 102. And this is an introductory video on one of the new scopes that you're going to be using this semester. You've got some other scopes which we'll talk about later. But this scope right here is the Olympus CKX41. And we've got several of them in the basement of Building S. But you, of course, won't be able to use them, unfortunately, until COVID's over. So I'm going to run through it here as I've set it up as a demo. And I'm going to then take some images with it, and you'll be able to use these images and process them at home. But I want to just go over what is this scope and what is the anatomy of it, the morphology. So it looks a little different than the Meiji. You might be thinking, why is it? Something's odd, but I don't know what it is. What's weird about it is, if you've never seen these type of scopes before, is that it's an inverted microscope. And that means the objectives are substage. Well, why would you do that? That seems silly. Well, main reason is that if you're doing cell culture, you've got cells, and those cells are on the bottom of a dish, and you need those cells on the bottom of the dish, and you can't turn the dish upside down because everything will go everywhere. Then you need to have the dish sitting on top, but then the objective's looking at it from below to check out those cells. So that's one of the main reasons. A lot of people have it in a cell culture lab. Well, they'll have one of these, and you can just check your cells and see if they're living or if they've adhered to where they're supposed to adhere to on, say, a slide. So that's one of the reasons for an inverted scope. What does that do, though? That makes things much more complicated. Instead of the light going through the sample, through the objectives, and then to your eyeballs in the Meiji, now we have this convoluted path where it's got to go down up here, down and then around. So I'm going to go through the morphology today on this scope, and it'll be an introductory of what is the scope. You will have a mastery exam. We'll have several of them. We haven't quite figured out what we're going to do, but the idea is you have to memorize the scope. What does it do? Why does it do it? How does it do it? Okay, let's do some basic stuff. Let's just follow the basic light path of, I just want to look at something on a slide. Okay, well, your simple button for looking at stuff on a slide is to turn on the light down here. This is the on button, but in this scope, this on button is really just for the bright field light, okay? Bright field light is this little black box, just does an LED light, and it goes through the condenser, and then this little slider here is a phase ring. We'll get to that later, but see, the whole thing could come out. It's a phase slider. It's got 3 different positions. We'll do that later. Then you can do a little bit of iris control or a little bit more control of the light, but eventually it comes out, shoots down here, and it hits the sample. Now your sample slide, for example this slide, it's upside down as I mentioned earlier. So you take your slide and then you've got to stick it upside down. And for those of you that have ever used oil, trying to stick oil on a slide and putting it upside down is quite a challenge. You won't have to do that. Okay, then we get to the stage here. The stage now can move this way or this way, so basically XY positions. This button here makes it go backwards and forwards. That's the top little ring. It's not a button. This bottom little ring goes this way. So you've got X and Y on this stage control here. You can't really see it, I don't think so well, but below it are a bunch of objectives, and there's an objective turret, and I'll go over that later on just what the objectives are. I'll try to get a better angle for you. So the light path goes down, goes through the objective, and then we've got some filter cubes down here with dichroic mirrors in them, and I'm moving it now, and again, maybe I'll just turn it down so you can see. How's that? Okay, hope you can see it. Can you see the BF and the B and the G? So what those are, inside there are several filter cubes, although for BF, which is bright field, it's really just one mirror, and the mirror, I think it's just one mirror. The mirror basically reflects everything, because you want bright field, you want all the light. The blue, the B, means that it's going to let in certain types of light for fluorescence, and G is for green. We'll get to that in a little bit. Okay. You've got it set on bright field now, which means all the light comes down through, goes into the objective. The objective is here. After the objective, it goes to this BF mirror, which is just at an angle, right? And the angle reflects the light into here. Then the light comes up this path, And now we have another choice point here, and it can either go into the oculars, and then you see it, woohoo, or it can go up to the camera, and you can take a pretty picture. So, hopefully a pretty picture. And you've got this little doodad here. This guy, you pull it out. Right now it's set at 100% ocular, so everything goes to the eyes, nothing to the camera. You pull it out midway, there's 3 points. Midway means it does both. So you can see the sample and you can get a picture of it. But you're missing some of the light into the camera, so it's not the best picture. And then once you get everything all set up, you pull it all the way out, then you take the picture. And the picture, 100% of the image and light is going up to this camera. That's bright field. Okay? If you wanted to take a picture with the camera, you would need to turn it on. So there's another on button here for the camera. This cord goes straight to the computer, and you'd be running something called Q imaging software or some other software. And many microscopes have this different option. It may not be a pull-out, but something says, do you want all oculars, do you want camera, or you want both, what do you want? And most microscopes with a camera do this. So that's basic bright field. Okay, what else did I miss? Oh yeah, you've got focus knobs. The big knob, like always, is the coarse focus, and then the little knob is the fine focus, okay? So now what it's doing is, you'll notice, it's not moving the stage. The stage is stuck. The Meiji, actually, this focus knob moved the stage up or down. So this coarse focus, if you can see, I don't want to go too far up because I don't want to smack the slide, but if I rotate it this way, sort of towards me, the objectives are going to move up towards the slide. So that's when you want to be careful because you don't want to scratch the lenses on the slide. And then if you move away from you, the entire turret, the objective goes down. So, can you see those? I think you might be able to see them. This, I'm holding it down here. There's the turret ring, and it moves the various objectives around. Right now, I have 4X on there. Okay? Let's see. What else do we need to know? If you're doing basic bright field, you also might do phase, because that's bright field. And so right now, I have the phase slider set on the lowest one, PHL, and you need to correlate these different phase rings. Can you see those? You need to correlate these different phase rings, which are really the same type of thing, the same different pattern, but they're just bigger or smaller. And oh, this one's a little off. That's strange. In any case, this one, small one, is for 4X, PHL, and then the middle one is for 10X and 20X, and then this one is for 40X. So you correlate this phase part, this phase ring in the slider, with what objective you're going to use. And so I'm going to put this in here. It just slides in easily. It's plastic. And I'm going to put it on 4X to correlate with my 4X objective. And we're ready to go for bright field. And now if I wanted to find a sample, I'd look in here and get my sample and focus it. Slowly go up until it's more or less in focus. And then use the fine. So it's easy to focus in on something. You always start on 4X because 4X means that A, the objective is also smaller and shorter, so you can't really hit it onto the slide, although you don't want to go all the way up anyway. But it's easy to see all of the sample, and then you don't have to worry about breaking anything. You can find where the sample is. When we get to fluorescence, it gets a little trickier. It's not so easy to find it with 4X sometimes because the light is so low. But you find the sample of 4X, and then you'd, of course, go up to 10X and 20X. And if you're doing phase, you'd have to move this for when you go to 10X. And I think that's it for bright field. All right. Now this baby also can do fluorescence. Woohoo! Fluorescence is fun. It's what I did in grad school. Make things glow in the dark. And what you're going to do is most of it's going to be very similar, except now let's follow the light source. Now the light source doesn't come from the top, go through the sample into the objective and get reflected up. Now the light source, and that's trans-illuminated light. Now the light source is this big black box back here. Inside here is a bulb, usually very expensive, usually very toxic, and usually very finicky and also fragile. Like really, like why would they design it that way? Well, now they have LED lights, and those are much more stable. They don't break as easily. They're not toxic. And they're also still just as expensive. But we have an older one here. It works really great. Once it's all set up and you're not really moving it around, it works really well. You're going to turn it on with this big box here. So this is the power box for the mercury light here, and there's a big cable going into it. So I'm not going to turn it on right now. Oh wait, yes I am, because I'm going to do fluorescence. Okay, you turn it on there. Starts making a bunch of noises. All right, you got to let it warm up. Takes a little while for everything to get going, and the lights won't appear. But basically, this is going to power the mercury bulb, and the mercury bulb it lets out many different wavelengths, but the wavelengths it lets out are very specific spikes in certain regions. So whereas bright field, we want a rich diversity of all the light that we can see. For fluorescence, we want distinct peaks in certain areas, and the mercury bulb gives us nice ones. Nowadays, they'll use lasers or very specific wavelengths and just stay green. But down here, we've got blue and green on this little slider, and what that's going to do is control which light comes out of the mercury bulb, because we only want a certain light coming out. That's called the excitation light. That light hits the protein that we're looking at, or the fluorophore, or the dye, and it makes it get all excited, which is why it's called excitation. And then, after it's been excited for a little bit, cools off, calms down, and it emits a certain light. And the light that is emitted, the wavelength is always a higher wavelength than the lower excitation wavelength. So if I want to excite with blue light, we've got two choices, blue and green. Let's say I'm going to put this slider in the middle. Of course, this sample is not a fluorescent sample, so I'm really just demonstrating this. I've got another sample later. Somewhere in here, okay, sort of funny, you got to push it in there until you get just the right, you can feel it stick a little bit. Now it's in the middle. What does that mean? Okay, this fluorescent cube, and you'll be quizzed on that, the fluorescent cube in there, it's going to only let in blue light from this mercury lamp. So all the light's coming in from the mercury lamp, and then the cube says, nope, filtered out, I only want blue to come in. That blue light comes through the filtering mechanism, passes through the dichroic mirror, which is very specialized to allow this to happen, and expensive. Then the blue light goes up through the objective, so the light is going into the objective, hitting the sample here. That's called epiluminescence. Sample's upside down. Light comes through the objective, hits it. The excitation wavelength, which is now blue, hits the sample. Sample gets excited, hopefully. Then it releases a higher wavelength. In this case, usually green is released from the blue excitation, and which is called green fluorescent protein. Then that emission light then goes down into the objective again, and now that specialized dichroic mirror, which is very, very expensive, and along with the filter cube, that then gets reflected up here and into the ocular, or like before, you can make it go to the camera. So that would be fluorescence for, you'd see green light, but blue light is what's coming through and exciting it. So if you're looking at the sample up here, you see blue shining everywhere, but then green is showing up here. Okay, and we'll go through more of that in the labs. Similarly, if you wanted to do green light excitation, you'd move the slider, you can't see my hand over here, I'm moving the slider down here, all the way to the left. Now it's on G for green. And the green one lets in only green light, so it filters out all the other light but green, which is now our excitation wavelength for a different protein. Green light does not excite GFP. We'll go into that. I know, it's confusing. Okay, green light comes in, hits the sample gets excited by the green light, then emits something higher, usually red or orangish. Red fluorescent protein, and then the red protein is fluorescing, and that light goes down and hits another dichroic mirror, because this is another filter cube, which is its own filter and dichroic mirror. That light goes through, the red light goes through the objective, bounces off the mirror, and then goes up this pathway into the light, and you can either, again, do the camera or do the oculars. Okay, those were the, I can see red in here. So, that is the basis of this. What you are going to do, first lab coming up, is you're going to start learning how to use this, and we're going to demo it for you, and we're going to do Project Zoom. And we're going to do it on histotech samples, and we're going to take pictures of it, mainly all bright field, although I think there's some fluorescence in there too. Then, what is lab 2? Lab 2 is me, so that was, lab 1 is going to be Mindy, module 1, and then, or not Mindy, Candy, and Dr. Mintz. And then I'm going to do module 2, which is going to be fluorescence. We'll be using this but we're going to be using the specialized type of sample that only glow in fluorescence, and you can't see the colors unless it's fluorescence, which is why I turned this on, because I'm going to do that in a little bit. And then there's going to be labs 3 and 4, and we're going to be using this for those. Although lab 4, I take that back, lab 4, we're going to start using another microscope, a Leica microscope, which has similar capabilities. Welcome to Bioscience 102.